Okay, hello to all of you. Let's discuss chapter number 16. And chapter number 16 is about DNA replication. So basically, the very first thing I would like all of you to know why DNA needs to replicate. DNA needs to replicate because cells needs to divide. So we have already talked about the cell division, and you have seen that the cell passes through different phases, and that's called cell cycle. And in the cell cycle, then there is one very long phase called interphase, and the interphase, there is a phase called S phase, that's called the synthesis phase, and the DNA duplication takes place in that phase. So basically, in mitosis, we have talked about that all the organelles, they are duplicated, and the DNA is also duplicated, and then it divides into half. So basically, in the interphase, there is a phase called S phase. In the synthesis phase, the DNA duplication takes place there, and that's what we are going to talk about in this chapter. We're going to talk about how DNA makes its copy, and we are going to talk about the proteins which are involved in that. And then, as you know, that DNA is double-stranded. The two strands of DNA, they are anti-parallel in nature. One strand goes from 5' to 3', another strand goes from 3' to 5'. They are anti-parallel in nature, and we are going to discuss that how these two strands, they make their copy. So there's a difference between the two strands because of their polarity, and we are going to discuss that. So there is little background about DNA, which I'm going to go over quickly, because this we have already discussed in chapter number 5 when we talked about the biomolecules. We know that James Watson and Francis Crick, these are the two scientists who actually deciphered the structure of DNA. The double helical structure of the DNA was actually discovered by them, and they also got a Nobel Prize for this. But it was actually the work of Rosalind Franklin, who, so you can see this is the model of, this is a model made by Watson and Crick, but the work was actually of Rosalind Franklin. So we already know what the structure of DNA is. We know that DNA is a polymer, and it is made of monomers called nucleotides, and every nucleotide has a pentose sugar, it has a nitrogenous base, and it has a phosphate group. So we have discussed all these things. So we know that in DNA, the nitrogenous bases are adenine, thymine, guanine, and cytosine. And we know that the two strands of DNA, they're antiparallel in nature, and they are connected to each other by weak hydrogen bonds, and they are connected to each other by complementary base pairing rules. According to these rules, A always links with T and G always links with C. And there's another scientist called Chargaff, and Chargaff's rules are very important, and there you can have some questions on the basis of Chargaff's rules. I have put some questions in your practice questions, the in-class quiz, which I post every week. Also, there are similar questions which you can solve on the basis of Chargaff's rules. They are in some mastering biology also, and you can expect those kind of questions in your quizzes as well as on your tests. So these are some of the Chargaff's rules. Let's discuss that. Erwin Chargaff reported that DNA composition varies from one species to the next. So every species, it has a different DNA composition. And these are the findings, two findings which became the Chargaff's rules. The base composition of DNA varies between species. As I said, in any species, the number of A and T bases is equal to the number of G and C bases. So basically, to understand Chargaff's rules, so let me go over the second point again. In any species, the number of A and T bases is equal. A and T bases is equal, and the number of G and C bases is equal. So which means that if you have a strand of DNA, how many A's it has, it will have the same number of T's. How many G's it has, it will have the same number of C's. So this rule applies to both the opposite strands as well as to one strand. Okay, this is very important thing to understand. How many A's are there in one strand, that strand will have the same number of T's. How many A's are there in one strand, the same number of T's it will have in the opposite strand. Okay, so in any species, the number of A and T's base is equal, and the number of G's and C bases is equal. So if one strand has 30 A's, it means the same strand, if we are talking about the same strand, it will have 30 T's. And if you talk about the opposite strand to which it is connected by hydrogen bonds, it will have 30 T's. So this is what is the main thing, and on the basis of that you can solve many questions. So this is the structure of DNA. We have already discussed. This is the pentose sugar, and the sugar in DNA is deoxyribose sugar. Then this is the 1' carbon here, and here is attached the nitrogenous base, and on carbon number 5 is attached the phosphate group. And this is the this is how these nucleotides are connected to each other by phosphodiester bonds. And if there is an opposite strand, it's going to be connected by hydrogen bonds. So this is showing you two strands of DNA. This is one strand of DNA. This is second strand of DNA. These two strands are antiparallel in nature. This strand goes from 5' to 3'. This strand goes from 3' to 5'. These are antiparallel. And you can see how the orientation of the sugar is flipped. So the sugar is, in this strand, the sugar is like this, and in this strand, the sugar is like this. And on the 1' carbon, here is attached the nitrogenous base. If in one strand it is T here, in the opposite strand it is going to be A here. If it is G here, it's going to be C here. If it's C here, it's going to be G. If it is A, it's going to be T. And the two strands of DNA, they are connected to each other by hydrogen bonds. And these hydrogen bonds are very weak bonds. And it has the big significance here that the two strands are attached to each other by hydrogen bonds because they are weak bonds. And you will see that in replication as well as in the gene expression, which we are going to cover next week, these two strands, they need to separate. And the two strands of DNA, they can separate only because they are connected to each other by hydrogen bonds. And the hydrogen bonds are the very weak bonds. So there's a big significance of the two strands connected to each other by hydrogen bonds. Okay, so these are the two strands of DNA. You can see that this is the way in different pictures in the book. You will see the two strands of DNA shown like this. It can be shown like this. It can be shown like this, or it can be shown like this, or like this. Okay, so now we are going to talk about many proteins work together in DNA replication and repair. So we are going to talk about many proteins, and first of all we are going to discuss the semi-conservative mode of replication of DNA. So basically, how does DNA make its copy? It makes its copy on the basis of a mode of replication, and that mode of replication is called semi-conservative mode of, it's called semi-conservative mode of replication. And what does that mean? It means that if this is the parental DNA molecule and it has two strands, you can see A is linked with T, C is linked with G, T is linked with A. So basically these two strands, they separate. Okay, these two strands separate, and each strand acts as a parental strand. Each strand acts as a parental strand, and on the basis of whatever is the sequence in the parental strand, a new daughter strand is formed. And from one molecule of DNA, two molecules of DNA are formed. So you can see these are the two strands of DNA. They separate. If it is A here, in the daughter strand which will be formed will be T. If it is C here, it will be G. If it is T here, it is A. And on the in the other strand, you can see in the other strand, if it is T here, in the daughter strand it is A, it is G, it is C, it is A, it is T, this is T, this is A, this is C, this is G. So this is the way the two strands of DNA, the two molecules of DNA, they are formed from one molecule of DNA. This is one molecule of DNA, and these are two molecules of DNA which are formed. And in the two molecules of DNA, one strand is old strand, which is a parental strand, and that parental strand acts as a template. And on this parental strand, a new daughter strand is formed. So from one molecule of DNA, two molecules of DNA, they are formed, and in each molecule of DNA, one strand is old strand and one strand is new strand, and that is why it's called semi-conservative mode of replication. And this concept of semi-conservative mode of replication was given by Watson and Crick. And there is one experiment which I just want to quickly go over, which actually, because at the time when Watson and Crick discovered the structure of DNA, there were many labs which were working on the structure of DNA. And after the structure of DNA was discovered, the next question was how DNA makes its copy, because it's important for cell division. So there were different models at that time. First is the conservative model, then the semi-conservative model, which was given by Watson and Crick, and the dispersive model. So according to the conservative model, which was given by another group of scientists, according to this model, when the DNA, from one molecule of DNA, when two molecules of DNA they are formed, from one molecule of DNA, when two molecules of DNA they are formed, one molecule is totally new and one molecule is totally old. That's a conservative model. And for dispersive model, from one molecule of DNA, when two molecules of DNA they are formed, the two strands which are formed, they have some part which is from old DNA and some part is new. Some part is old, some part is new. And the same thing is for the second molecule of DNA. Some part is old, some part is new. But according to the semi-conservative model, the two strands separate, and when the two molecules of DNA are formed, one strand is new and one strand is old. So how it was, so these were the three models prevalent at that time. But how do we know that Watson and Crick's model was correct? There was the scientist called Michelson and Staw. These are the two scientists, Michelson and Staw, and they are the ones who actually gave the semi-conservative. They are the ones who did the experiment and proved that the Watson and Crick's model was correct. So let's see what the experiment is. So basically, what they did, they grew the bacteria. Okay, so as you know, if you have studied microbiology, that we can culture the bacteria in the lab, and for culturing the bacteria, we need to grow the bacteria in the culture. So what they have done here, they have so for the growth of the bacteria needs carbon, bacteria needs hydrogen, bacteria needs oxygen, and bacteria needs nitrogen. So basically, this is the medium. So this is the medium in which the bacteria is grown, and in this medium there is nitrogen, and this is nitrogen 15. So it's a radioactive nitrogen. So it means that the bacteria which is growing in this medium, the DNA which is there in the bacteria will have nitrogen 15. The DNA we just talked about, it has nitrogenous bases. There is a nitrogen. So it means that the nitrogen which is there in the bacteria, when the bacteria is cultured in this medium, its DNA will have nitrogen 15. And what they did, they took this bacteria and they cultured this bacteria from this medium to another medium in which the nitrogen is nitrogen 14. The nitrogen is nitrogen 14. And after that, they kept growing these bacteria from in the medium which is nitrogen 15. And what they did was they took the bacteria which was growing in this medium. They took the bacteria which was growing in this medium and they isolated its DNA. So the DNA which is coming from this bacteria, it should have nitrogen 15. And then they took this bacteria and they put it into this medium in which the nitrogen is nitrogen 14. And then again they did the second cycle of replication. So what they did, they after the first cycle of replication, they collected the bacteria and they prepared DNA from that, and they put it on this gradient. Okay, this is a kind of a density gradient. It has some material and it separates the material or the molecules or the DNA, we can say, on the basis of the density. So what they found, that when the bacteria which was being grown in this medium, when it transferred to this medium, they collected the bacteria, they prepared the DNA, and they passed over this column, and they found only one band. So let's see, on the basis of these results, which model is correct. Because they got only one band, on the basis of conservative model, that from one molecule of DNA, when two molecules of DNA they are formed, one molecule should be totally new and one should be totally old. It means that there should be two types of DNA bands should be there. So which means that according to the conservative model, on the density gradient there should be two bands, but they were getting one band. So it means that conservative model is wrong. So on the basis of dispersive model, because on the basis of this model, the two molecules of DNA which are going to be formed, they should have some patches of old DNA and some patches of new. So which means that on the basis of that, they should get one band. It means, yes, maybe dispersive model is correct. And on the basis of Watson and Crick semi-conservative model, two strands they separate, the two strands of DNA they separate, and and on the basis of the sequence in the old strands, new strands are going to be formed. So on the basis of semi-conservative model also, the two molecules of DNA, they are going to be formed. They will be same basically, because one strand is old, one is new. So it means that on the basis of this model also, they should get only one band. So which means that from the results of the first cycle of replication, they could rule out this model, that this model is wrong, but these two models are correct. So now the next question is to know which model is correct out of them. So what they have done, they have passed this bacteria from this medium to the medium where there is nitrogen 14. And what did they find here? What they found here, they found two bands. Okay. Now on the basis of dispersive model, they should have got one band, but they're getting two bands. And on the basis of semi-conservative model, they should get two bands and they're getting two bands. So that is why the semi-conservative model is correct. Now let me explain you how semi-conservative model is correct. Let me go here and on the board and show you. Okay, so now as we have seen, the bacteria which is in the medium, which is being grown in N15 medium, so it will have two strands of DNA which will have nitrogen 15. Okay, so now when this bacteria is passed to the medium which has nitrogen 14, according to the semi-conservative model, these two bands are going to separate, right? So this is a parental strand. It will have nitrogen 15. This will also have nitrogen 15. And because the new strand is going to be formed, and that new strand is going to be formed in the new medium, which means the source of nitrogen is going to be the new medium. It means that the new strand which will be formed will have nitrogen 14 in it. Okay, the same thing is going to happen here. So on the basis of the first replication, you can see the two molecules of DNA are formed. Two molecules of DNA, they are formed. One strand has nitrogen 15, other strand has nitrogen 14. In the second molecule of DNA, one strand has nitrogen 15, second strand has nitrogen 14. So these two molecules are same. So that is why on the density gradient you got one band. Now in the second cycle, what will happen? Now if we talk about this molecule, what will happen? These two strands will separate. Okay, these two strands will separate. So this has nitrogen 15. Okay, and this is the second strand. This will also separate. This is nitrogen 14. Now this bacteria which has this kind of a DNA, this bacteria is going to grow in the medium with nitrogen 14, which means the new strands which are going to be formed will be having nitrogen 14. And this will be also nitrogen 14. So, and the same thing will happen to this molecule. So which means that when in the second cycle of replication, when this molecule is going to make its copy, these are the two kinds of DNA molecules will be formed. This molecule is having one strand having nitrogen 15, second strand has nitrogen 14. And this is second molecule of DNA. This is second molecule of DNA which has both strands having nitrogen 14. So in the second cycle of replication, the two molecules of DNA which are formed, they are not same, just like the first cycle, they are different. One molecule is a hybrid, another is pure, which means both strands are same here. So one band, so that is why on the density gradient they saw two bands. One band is for this, one band is for this. Now this band is exactly the same band which they got for the first cycle because it's the same. The second band they're getting is because of this molecule, and this molecule has both the strands N14. N14 is a lighter isotope of nitrogen. So this band shows at a lower position, and that's what they got. Oops, sorry. I'm sorry. Okay. Let's go back here. So if you see here, this is the way they're getting. Okay. So they are getting this one band is same here, which was there in the last cycle also. So this is the hybrid molecule, one old strand, one new strand, one having N15, another having N14. And this is the second band they're getting on the density gradient, which is lighter band, and this is because this molecule has both strands have N14 in that. It is less dense, that's why you see it here, because anything which is going to be dense, it's going to be coming at the bottom. Okay, so this was the experiment which showed that the semi-conservative mode of replication of Watson and Crick is correct. Okay, now we're going to talk about how exactly DNA replicates. In this, we are going to talk about some of the enzymes. Okay, and first of all, we are going to talk about how the DNA replicates in prokaryotic cells and how the DNA replicates in eukaryotic cells. Prokaryotic cells like bacteria and archaea, they have basically, bacteria, they have only one single chromosome. So the DNA is circular, and in the eukaryotic cells, they have linear chromosomes. The DNA is linear. So basically, the DNA replication starts at one point in DNA, that's called origin of replication. So origin of replication is that point where the two strands of DNA, they start separating. So you can see how the two strands, they have started separating, and then this then the replication bubble, it becomes big, and the two strands, they are copied, and then from one molecule of DNA, two molecules of DNA are formed. And I'm going to discuss this every term in detail coming forward. I just wanted to tell you how basically how the replication of DNA takes place in the prokaryotic cells. So this is the circular DNA, and the eukaryotic cells, there's a linear DNA. In the prokaryotic cells, there is only one origin of replication. In the eukaryotic cells, because they have long linear chromosomes, they have multiple origins of replication, and they have multiple replication bubbles. Okay, and the DNA replication takes place at multiple places, and then how the whole chromosome gets replicated. So this is prokaryotic, this is eukaryotic. So it starts from here, and then the two strands, they make their copy, and the two molecules are formed. And we are going to discuss in detail about how it actually takes place and all the terms and all that. So basically, first of all, we need to know some terms. So first of all, what is a replication fork? At the end of each replication bubble, so what is the replication bubble? So this is a replication bubble. Okay, so this is the place. Replication bubble is that region where the replication is taking place. So you can see how the replication bubble is being formed here. And so this is the replication bubble. This is the replication bubble, and this is the replication fork. So what is the replication fork? At the end of each replication bubble is a replication fork. It's a Y-shaped region where new DNA strands are elongating. Then there's the involvement of an enzyme called helicases. Helicases are those enzymes that help in the opening of the, it helps in the separation of the two strands of DNA. So DNA is double-stranded. For the replication to take place, first of all, the two strands need to separate. The two strands, they need to separate, and these two strands, they separate by the enzyme called helicases. Then when the two strands is separate, these two strands should not come back. These two strands should not come back. And these two strands, they do not come back with the help of these proteins called single strand binding proteins. When the two strands they separate, the two strands become single strands, and there are some proteins which are called single strand binding proteins. They go and bind to these two strands so that these two strands do not come back. And there's involvement of an enzyme called topoisomerase. It relieves the strain of twisting of the double helix by breaking, swelling, and rejoining the double strands. So when the DNA strands, the two strands, they're opening, it's just like a spring. You can just imagine like a spring. If you start opening a spring, there will be a lot of tension ahead of. There will be a lot of tension ahead when the two springs are opening. The spring is going to become very tight, and the same thing happens with DNA also, because DNA is very, very super coiled, but because of the involvement of these enzymes called topoisomerases, they relieve the strain of twisting of the double helix. These are the enzymes which help in that. So this is basically, you can see there's the involvement of the enzyme called helicase. Helicase basically helps in the separation of the two strands of DNA. So the two strands they separate with the help of helicases. And then these are the single strand binding proteins. They go and bind to the two strands so that they don't come back again. And then topoisomerase, which helps in, it removes the tension which is going to be there when the two strands of DNA, they are separating. And then there is an important enzyme involved called primase. Now what is a primase? Primase is an enzyme which helps in the formation of a RNA primer. Now what is the RNA primer? RNA primer is a short stretch of RNA which is made, so it's a short stretch of RNA which is made before the DNA can be replicated. The reason for, the primase is an enzyme which helps in the making of RNA primer. So there is an involvement of a very important enzyme called DNA polymerase. DNA polymerase 3, DNA polymerase 1, we will talk about these two enzymes, DNA polymerase 3, DNA polymerase 1, what exactly their functions are, what exactly the contribution is. We're going to discuss very shortly. But first of all, let's understand what DNA polymerase is. DNA polymerase is an enzyme which actually helps in the building of the daughter strand. It is the function of DNA polymerase which makes the new strand or the daughter strand of DNA. And DNA polymerase makes the new strand on the basis of complementary base pairing rules. If in the parent strand there is A, the function of DNA polymerase is to bring T. If there is a nitrogenous base G, the function of DNA polymerase is to bring C. If it is C, G. If it is T, A. That's the function of DNA polymerase. DNA polymerase makes the new strand of DNA. But DNA polymerase works in a specific direction. Okay, what is its direction? It works in 5' to 3' direction. Let me go back to the board and show you here. So it works in 5' to 3' direction. Okay, so which means that something very important to know about DNA polymerase is it needs 3' end. Okay, there is a drawback or a shortcoming of this enzyme, that it cannot initiate the process of DNA polymerization. It can elongate. So that is what primase is doing. Primate is putting a small RNA primer. Okay, and what that RNA primer is doing, RNA primer is nothing but a short stretch of DNA. So you need to know at this point what is RNA, what are the differences between DNA and RNA, which we have already discussed. So the short stretch of RNA is going to be like this, 5' to 3'. Okay, and this RNA primer is going to provide this free 3' end to DNA polymerase, and that's what is the requirement of DNA polymerase. DNA polymerase cannot initiate the process of DNA polymerization, but it can elongate. So that is why there is an involvement of another enzyme called primase. Primase makes RNA primer, and this RNA primer is made in this direction, 5' to 3'. and it provides free 3' end. If you go back and look at the structure of the nucleotide, you should know that on the 3' end there is a OH group, and that is what is required by DNA polymerase, because the function of DNA polymerase is to bring a new nucleotide on the basis of complementary base pairing rules, and that nucleotide will be connected to the nucleotide which is already here, and for that it needs free 3' end. Only then a new phosphodiester bond will be formed between the old nucleotide and the new incoming nucleotide. So this is what is the requirement of DNA polymerase. And that requirement is fulfilled by this primase, because it makes an RNA primer, and this RNA primer is made in this direction, 5' to 3', and DNA polymerase needs this. So once it is made, DNA polymerase is ready, and it is going to make a new daughter strand of DNA, and it has got what it requires. If it is A here, it's going to put T. If it is T here, it's going to put A. If it is G, it's going to put C. If it is C, it's going to put G here. Okay, that's the way it works. Okay, so this is all what I just said. It's written here. You can go over it. Okay, so this is showing the process of DNA polymerization by DNA polymerase. So you can see that this is the old strand, and this is the new strand which is being made. This is the old strand. If it is T here, it is A here. If it is G here, it's C here. If it is C, it's G. Now let's see how the new incoming nucleotide is being added to the daughter strand. So if it is A here, the function of DNA polymerase is to bring T, and these nucleotides, they have 3 phosphates with them. Okay, so the function of DNA polymerase is to add the T to this strand, and for that it needs 3' end free, where there is a OH group, and it is going to it's going to link this T with this nucleotide, and that forms the phosphodiester bond. And the nucleotide which is attached in the chain, in this strand, has only 1 phosphate. So the 2 phosphates which were there in the incoming nucleotide will be released as pyrophosphate. This is the way how DNA polymerase functions. Okay, now very important thing here is DNA is double-stranded, and we already know that the 2 strands of DNA, they are antiparallel in nature. Now the question is, now one of the strand can be made, another strand cannot be made, right? Let me go back here and show you on the board what I'm talking about. Okay, let's erase everything. Okay, 2 strands of DNA, they are antiparallel in nature. One strand is 5' to 3'. Oops, 3'. This strand is 3' to 5'. Okay. 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 So these are 2 strands of DNA, right? They are antiparallel in nature. They have this kind of a polarity. Okay. Now, if this strand is 3', and we know that DNA polymerase needs a primer, and that primer is made by primase. For this strand, the primer will be made like this. Let me make a primer with different color. Okay, the primase is going to make primer like this, right? If this is 3', on the primer, this will be 5'. Remember that this is a process of making 2 strands of DNA. Remember that the 2 strands of DNA will be antiparallel, because this new strand is going to be a daughter strand, and these 2 strands are going to make a new molecule of DNA. So this is 3', the new strand will have 5' here, and this side will be 3'. Okay, now what does DNA polymerase needs? DNA polymerase needs free 3' end. So now, so this is RNA primer. DNA polymerase will come, and it will make the new strand of DNA here. Because it needs 3' end, it is getting it, it will make a new strand. All good, right? Now, there's a problem with the opposite strand. And what that problem is? Now, if the RNA primer is made, this will be 3 prime, this will be 5 prime. Okay. Now can DNA polymerase come here and make the DNA strand? No, it cannot. Why? Because on this side, the free end is 5 prime. And what is on the 5 prime? A phosphate group. What does DNA polymerase needs? 3 prime end. It needs hydroxyl group. It's not there. So DNA has 2 strands. They are antiparallel in nature. Then how does the second strand is made? How does the second strand is made? The second strand is made in a different way. Okay. Now let's talk about how the 2 strands of DNA they are made. Now the strand, this strand, which can be made like this. Okay. This strand can be made like this, no problem. This strand is made continuously, and this strand is called leading strand. The opposite strand cannot be made. Opposite strand is made because DNA copies itself. The opposite strand is made, but it is made in a different way. And the opposite strand, it cannot be made continuously. It is made in fragments, and those fragments are called Okazaki fragments. Okazaki was a Japanese scientist who actually discovered this process. He is the one who found out that the opposite strand of the DNA is made in fragments, and these fragments were called Okazaki fragments. Okay, and the second strand of DNA is called lagging strand. Okay, and the lagging strand is made in a little different way. Okay, and it involves the 3 enzymes. It involves DNA polymerase 3, it involves DNA polymerase 1, and third enzyme which is involved is DNA ligase. Okay, now I'm going to show you how the 2 strands of DNA they are made. Okay, okay, so now let's make a DNA structure here, and now we are going to make use of all DNA bubble, origin of replication, and all that. Okay, suppose this is the 2 strands of DNA. Okay, suppose these are 2 strands of DNA. First of all, let's write about the polarities. Okay, now polarity of DNA is very, very important. So this is 5 prime, this is 3 prime. Okay, this is 1 strand of DNA. If this is the opposite strand, this will be 3 prime, this will be 5 prime. Okay, now this is origin of replication. Okay, so origin of replication is that point in DNA where the 2 strands of DNA they start separating. Okay, that's called origin of replication. Okay, so if this is 5 prime, now let's use a different color. If this is the, so basically here the 2 strands are separating. If this is the 5 prime, then this side of this strand is 3 prime. If you look from here, okay, on this side, if this is 5 prime, then this side is 5 prime on this strand. Same thing here. If this is 3 prime, this side of the strand is 5 prime, and this side is 3 prime. Okay, now let's talk about how they are going to be replicated. Okay, now let's use different color. Let's use green. Okay, for this strand, okay, for this strand, if this is 3 prime, the primer which will be made by primase will have here 5 prime, correct? Okay, let's first make the primers. If this is 5 prime, this is 3 prime, the primer which will be made on this side will be having 3 prime, and this is going to be 5 prime. Okay, for this side, if this is 5 prime, the primer will have, this will have 3 prime, this will have 5 prime. For this side, if this is 3 prime, this is going to be 5 prime, this is going to be 3 prime. Okay, now let's use this color. Now if this primer is having 5 prime on this side, 3 prime on this side, DNA polymerase needs 3'. So this strand can be made continuously. What about this side? This is 3', this is 5'. Can this strand be made continuously? No, it cannot be, because DNA polymerase is not having free 3' end. So this strand is going to be made discontinuously. It will be made in fragments. How exactly it's going to be made, I'm going to come on that. Let's go over the opposite strand and see. So this side, 5' to 3', this can be made continuously. This side cannot be made continuously. It's going to be made in fragments. So this strand is going to be leading strand. This is also leading strand. This is going to be lagging strand. This is also going to be lagging strand. Okay. So this side, this strand or this side, basically, this is leading. It can be made continuously. This can also be made continuously. This is also leading. This strand, this part of the DNA cannot be made continuously. It's going to be made in fragments. It's a lagging strand. This side is also lagging strand. Now, how they are made in fragments? Remember that DNA polymerase works only in 5' to 3' direction. This is a very, very important direction, 5' to 3'. DNA replication takes place in 5' to 3' direction. Transcription takes place in 5' to 3' direction. Translation also takes place in 5' to 3' direction. Okay. So now let's see. DNA polymerase works in 5' to 3' direction. So the DNA polymerase is going to work in this direction. Okay. And this strand is going to be made in chunks. It's going to be made in fragments. It involves the enzyme DNA polymerase. So because the DNA polymerase works in this direction, first of all, the primase is going to make a RNA primer here. Okay. It's going to make multiple primers. It's going to make one primer here. It's going to make one primer here. Okay. It's going to make multiple primers. And then DNA polymerase 3 is going to elongate it. Then DNA polymerase is going to elongate this primer. It's going to elongate this primer. Okay. Then comes the function of DNA polymerase 1. DNA polymerase 1 is going to come and remove these primers, because these enzymes, they not only have polymerization capability, they also have a nuclease activity. They can also remove. So DNA polymerase 1 is going to come and remove these primers, and it is going to fill these with DNA. And then there will be a gap between, so suppose this is the, let's make the purple color. So I'm just explaining what is happening here. If this is the DNA fragment which is made by DNA polymerase 3, then DNA polymerase 1 will come and it will remove the RNA primer, and it is going to make a new strand here. Okay. So this will be the strand made by DNA polymerase 1. This is the strand made by DNA polymerase 3. There will be a nick. There will be a gap between these two strands. Then comes the role of the third enzyme called DNA ligase. It is going to come and seal this gap. Okay. This is the function of DNA ligase. It is going to come and seal this gap. It's going to connect these two strands of DNA. It's going to connect these two strands of DNA. And how does it connect? By making the phosphodiester bonds. So this is the way how the two strands of DNA, they are made. Okay. One strand is made continuously. Second strand is made discontinuously. The strand which is made continuously is called leading strand. The strand which is made discontinuously, it's called lagging strand. And there is the involvement of so many enzymes in this process. Okay, so this is basically showing you, this is what I just drew, and you can see this side is leading, this is lagging. And all these enzymes are involved in this. So this is all the machinery. So we call it a DNA replication machinery. It's all these proteins are there. So this is the origin of replication. So first of all, there's the formation of a RNA primer, which is made by primase. Then the DNA polymerase is going to come and it's going to make the new strand. And helicase is there, single strand binding proteins are there, topoisomerase is there. So all these enzymes, they make the machinery, replication, DNA replication machinery. So now let's exactly talk about how the lagging strand is made. As I just explained on the board, that you can see that it is made discontinuously, it is made in fragments, and RNA primase is going to come and make multiple primers. Okay, it's going to put a primer here, it's going to put the primer here. Once the primer is made, DNA polymerase 3 is going to come, and it is going to it needs a 3' end. So this 3' end, which has a hydroxyl group, is given by the RNA primer, and DNA polymerase 3 is going to come and it is going to make a Okazaki fragment. Okay, once the DNA polymerase 3 meets the second primer, it's going to leave the DNA. Okay, so you can see here, here's the primer, here's the second primer. DNA polymerase 3 will make the new strand of DNA here, it will make the new strand of DNA here. Okay, and once it comes across the second primer, it's going to leave the DNA. So this is the way how the lagging strand is made. So it means that the lagging strand is a mix of DNA and RNA. But when the DNA makes its copy, there is no RNA. So how that RNA is removed? That RNA is removed by the enzyme called DNA polymerase 1. It will remove, first of all, the RNA. It will remove the RNA and it is going to substitute the RNA with DNA. And then comes the role of the enzyme called DNA ligase. It is going to seal the gap which is between the DNA fragments, because one DNA fragment is made by DNA polymerase 3, one is made by DNA polymerase 1, and that's what DNA ligase does. So this is the way how the lagging strand is made. So this is what it is. So this is the complete DNA replication machinery. So one strand is leading strand, it is made continuously, and the second strand is the lagging strand. This is the list of all the proteins involved. You must know all these proteins because you can have a lot of questions on the basis of this. So this basically talks about how the DNA makes its copy. And now suppose there is, although the DNA replication process has a lot of fidelity, which means there are very less chances of mismatching happening because these enzymes, they have a proofreading activity. So if there is A, only T should be attached. If it is G, C should be attached. But still sometimes, if the mismatch happens, these enzymes, they can remove the mismatched nucleotide and put a correct one. But sometimes still there is a damage to the DNA. All these things can happen. In that case, we have a repair mechanism in our DNA, and it is called nucleotide excision repair. In this, again, there is a set of enzymes involved. In this process, first of all, the nucleus, and if suppose there is a damage to the DNA here, this part of the DNA will be removed by nucleus, and this part will be this part of the DNA, because it's removed from here, will be filled by DNA polymerase, and then there will be a gap between the new strand and the old strand that is going to be sealed by DNA ligase. So this is the way how the replication, the repair process, it works. Okay. Now, as we have just talked about how the lagging strand is formed. Now there is a question which comes, how the lagging strand is formed. See here, as we discussed, lagging strand is made in fragments. There are multiple primers, right? So we talked about, here is one primer, here is one primer. So this part of the DNA is made by DNA polymerase 3. After that, DNA polymerase will come and it will remove this primer. It will remove this primer and it is going to make, it's going to put DNA strand here, DNA nucleotides here. Important thing about DNA polymerase is it needs 3' end. So when this primer is removed, the 3' end is given by this part of the DNA. But what about when this primer is removed? What about this? When this primer is removed, when this primer is going to be removed, how this part is going to be filled? Because This side of the DNA is 5 prime. 3 prime is on this side. For filling this part, for filling this gap, DNA polymerase needs 3 prime end. It's not going to get it. Why? Because 5 prime is on this side, 3 prime is on this side. To fill this gap, there is no 3 prime end. So this part of the DNA is not filled. So which means that when the two strands they are made, the lagging strand always loses some part of the DNA, because the end of the DNA cannot be filled, right? So it means that lagging strand will always have some part removed from the ends, and then when this, so the DNA molecule will be formed like this, and when this is going to go to the second cycle of replication, again same thing is going to happen. The lagging strand is again going to have some part missing from the ends. So it means that the way the DNA replicates, the lagging strand will always have some part of the DNA removed from its ends, and that is true. That's the way the DNA replicates. That's going to happen. So does this mean that when the replication is taking place, some part of the DNA is always removed? If it is that is happening, yes, that's happening. If that is happening, it means that in every cycle of replication, our cells are losing some genes. Yes, the DNA is getting eroded from the ends. It is getting removed from the ends, but we are not losing some genes. The reason is, if you have heard about telomeres. What are telomeres? Telomeres is basically a short stretch of some random DNA sequence. Okay, so at the end of our chromosomes, we have some random non-specific DNA sequence which does not code for any gene. So which means that yes, in every cycle of replication, in every cycle of DNA replication, some part of the DNA is getting removed, but we are not losing some of the important genes, because at the end of the chromosomes there are telomeres. They do not code for any gene. We also call telomeres like caps. They are caps on the chromosomes. So every normal cell can only undergo a specific number of cycles of replication, because once the telomeres are removed, the cell cannot further divide. The cell has to die. So that is why every normal cell has a finite lifespan, and that lifespan is determined by the length of the telomere. Once the telomere is gone, the cell has to die, because after that the cell will start losing some of the important genes. Okay, so that is why every normal cell has a finite lifespan. If we talk about cancer cells, they are immortal. They keep on dividing. How can they do that? Why they don't have finite lifespan? Because they have an important enzyme in them called telomerase. And what does this telomerase does? It catalyzes the lengthening of telomeres in them. Okay, and the same enzyme is also present in the germ cells. Okay, so that is why. So you know that we have two different kinds of cells in our body. We have somatic cells, we have sex cells or the gametes. These gametes also have this enzyme called telomerase, and it catalyzes the lengthening of the telomeres. So that is why these germ cells, they don't die. That's why they are able to pass the information from generations to generations. And the same enzyme is present in the cancer cells. So this enzyme helps in the, it catalyzes the lengthening of the telomeres. So that is why in the cancer cells, these telomeres, they are not eroded. So that is why this regulation is lost by the cancer cells. That is why cancer cells can keep on dividing, because the telomeres are not eroded. Why? Because they have overexpression of this enzyme called telomerase. And there's a lot of research going on currently on this enzyme, how we can download how we can down-regulate the expression of this enzyme so that the cancer cells can have a finite lifespan, so that they can also die. That's what it is. Okay, this picture, if you see, this is showing, this is a fluorescent, this is a picture taken by fluorescent microscope, and these are showing these points which you see, which are being lit. This is showing fluorescence because this is a picture taken by fluorescent microscope, and these points are basically showing the telomeres. So you can see how the chromosomes are and how at the ends of the chromosomes there are telomeres, and they're also called caps on the chromosomes. Okay, and the last topic we are going to discuss in this chapter is that how does this DNA, we know that DNA is carrying millions of genes on these chromosomes, right? So DNA is a very, very long molecule. So our cells, we cannot see them with the eyes, right? We have to use a microscope. So in the cell, there is a nucleus. Nucleus is even smaller. How does this all long molecule of DNA gets fit into the small nucleus? So there is a packaging pattern, or there's a way how does this DNA gets packed inside the small nucleus. So basically, the DNA is wrapped very, very tightly on the proteins called histone proteins. So we have already discussed the charge on DNA is negative, and it is because they have phosphate groups. So these histone proteins, they are made of some amino acids which have positive charge on them. Arginine, histidine, these are some of the amino acids of which these histones are made, and they are positively, these amino acids, they have positive charge on them. This DNA, which is negatively charged, it is very, very tightly wrapped over these histone proteins because of the opposite charges, and they form nucleosome. So you can see this is your DNA, this is DNA helix. It's 2 nanometers in the diameter, and these are the histones, and these are the histones, and you can see how the DNA is wrapped over these histones. Okay, and when the DNA is wrapped over the histone, it's called nucleosome, and the diameter becomes 10 nanometer. You can see how it is becoming thick. And then these nucleosomes, they further coil, they further fold, and then they form chromatin. And the chromatin fiber is around 30 nanometers in the diameter. And then it further folds, further coils, forms loops, and it further becomes around 300 nanometers in the diameter. Okay, so this is what the basically the chromatin is, and we have already discussed in our previous chapter that when the cell is not dividing, it is like a dense mass, and dense mass is basically of the chromatin, which is DNA wrapped over the protein, and it is diameter is around 300 nanometers. And when the cell enters into mitosis, when the cell enters into division, it becomes more thick, it becomes more condensed, and it attains the shape of chromosome. And the chromosomes, they are duplicated, and they have 2 sister chromatids. So the diameter of each chromatid is around 700 nanometers. So this is the way how, and if it is a replicated chromosome, so there are 2 chromatids, the diameter is around 1400 nanometers. So basically this is the way how DNA is packed inside the small nucleus. This is the packaging pattern of DNA. And in the chromatin, there are 2 kinds of chromatins. This is basically just showing that how the different chromosomes, they have specific position. So this is chromatin, these are chromosomes. Okay, so you can see like this is, suppose this is your chromosome number 3. This chromosome number 3 is here. Okay, this is chromatin, and these are chromosomes. And in the chromatin, there is a loosely packed chromatin, it's called euchromatin, and then there is a tightly packed chromatin, it's called heterochromatin. So usually at the regions like centromeres and telomeres, there the chromatin is very highly condensed. So that is called heterochromatin. So wherever the chromatin is very tightly packed, their expression of the genes cannot take place. There are many mechanisms, there are many modifications which take place to the histones, and because of which this chromatin becomes loose, and then the genes can express. And the gene expression we are going to talk in the next chapter. So in this chapter we have discussed about how DNA makes its copy, In the next chapter, we are going to talk about how DNA expresses itself, how genes express and they form proteins. So that's a topic for the next chapter. So that's all in this chapter. If you have any question, please let me know.